fine. So I'm working in CERA in Grenoble, south of France. I'm working since 10 years on different type of microscope. And microscope, there are more and more algorithms into this microscope, and nowadays there are more and more deep learning. So this talk, I think it's a nice follow-up of the talk from this morning from Christoph Zimmer, because Christoph, he went very much into the details of deep learning in a very didactic manner. And I will give you more an overview. I will focus on the applications, and especially the application in the field of microscopy. And in the end of the talk, I will discuss in particular the work that we are doing in Grenoble in this field. So I will start with a short introduction, an historic point of view, to go from cell observation to the quantitative analysis. Can I remove the mask? Yes. Thank you. The part one, sorry, I have to get used. The part one will be the introduction of deep learning into the field of microscopy. It has started 5 years ago, so it's not that long, but you will see that over 5 years, it has changed a lot, the field of microscopy. And in the end, I will give you some insight on the work that we are doing in Grenoble, and it deals with the new representations that are brought by deep learning. So microscopy, it all started in the 17th century in Delft, the Netherlands, with Anthony Leeuwenhoek. And the microscopy, it was already a sample, some optics, and a scientist. The nice thing at that time that every observation was a discovery. That means every time they put a sample under a microscope, it was a discovery. And nowadays, it's very hard to make any discovery, I would say. I'm not sure myself I will do any discovery in the time of my career. I did not so far. There are some years remaining, but it's very hard to make any discovery. In that time, they discovered the cement, they discovered the cells, they discovered the microbes, they discovered the bacteria. Just every time they make an observation. That was quite a nice time for that. In the 70s, we put camera on top of the microscope so that we were able to make nice pictures. And the pictures, they became very nice in the 80s, at the point we put fluorescence on top of the cells. So far, it was... we were looking at transparent sample from that point, from that years in the 80s. It's not possible to put colors on the distinctive organelle of the cells. Here, in blue, you have the nucleus. In green, you have the microtubules. So this is in the years in the 80s, but we are still in the phase of observation. We look at the cells. We are not yet measuring, and this comes with the algorithm. This is another example of fluorescence microscopy to show you that this is an observation. It's a very fancy observation because it is in 3D, and it's 3D plus time. And you see here the very early moment of embryogenesis. I don't know if it's a zebrafish or Drosophila, but these are the very first moment. This is still observation. You are able to look in very sharp details all the cells, the nucleus, the membrane, and so on, but you are not measuring anything yet. It's not about quantification yet. Quantification arrived in the 80s, in the 90s, thanks to algorithms. And now we are in the year 2018. Here you are looking at the development, cell by cell, of a Drosophila. And it's not only observation here, it's quantification, because you are able to make a track of every single cell, and you are able to distinguish the nucleus in every single cell, and you are able to track every single cell, so that you are able to follow the full development of the early moment of the embryo, thanks to an algorithm. And now you are making quantification, now you are making measurements. And actually, this has been awarded as the innovation of the year 2018, the ability to look in an automatic manner to a very large number of cells thanks to the algorithm. On this picture, what you see in magenta is the cell division. So you see that there is a wave that's going from left to right with the division. And this is done only through the algorithm. So at that point, we are able to make a measurement, automatic measurements. And we are in the year 2018. So we spent a few centuries here, and we will go to 2021. So this is the video for the award in 2018 by the Science magazine. You can see what it's possible to do, and the color is obtained by an algorithm. Here it's the observation of the fluorescence. And this is an algorithm who is pointing out a different type of cells through the evolution of the embryo. So this is 2018. And nowadays, 2020, 2021, we're able to reconstruct in sharp details the full, not the full brain, but I guess a part of the brain of a Drosophila. So now it's not me, it's not myself, it's the humanity, but in principle, the institute here in the Howard Hughes, you will see that there are many slides. This is an institute in the US. They are very good. They get a Nobel Prize every year about. And they, so not we, but they are able to compute in 3D the brain of a Drosophila. So it contains 25,000 of neurons. Every neuron has been detected by an algorithm, and it can distinguish about 4,000 types of different neurons. This is done fully automatically. At first, you need to, these are electron microscope images. So these are slices of the brain of the Drosophila that are brought to this electron microscope. There are 10 of them. They are running 24 hours a day. So you need this kind of microscope to produce this image. But what's important here is to analyze this data. These are not anymore regular algorithms. To analyze this huge data, you need deep learning. And the whole pipeline that has analyzed this data is made of deep learning. And to have a good deep learning, this is it. One of the challenge was to make the segmentation of the neuron in this 2D image. Maybe a child is able to make the colorization, but it's not an easy task for a computer. You remember this morning when Christophe just showed you the gray level value. You will not. It's very hard to just from the gray level value to define rules and to define a code to make this computation. And nowadays, this is done by deep learning. So my point here is that till a few years ago, we increased the observation, we increased the details, we increased the amount of information in the data, but we were not able to process it as we are able to do it. And this is a very nice example to show that deep learning nowadays is able to make the segmentation in 2D. So the neural network goes from this image to this segmentation image, where every color corresponds to a given neuron. And then the deep learning, the neural network, is also able to follow this neuron all along the brain, over millimeters, maybe centimeters. So you see that on one plane, you are able to make the segmentation of the neuron, but next, it is able to follow up this neuron in 3D. And this is done fully automatically. The point here is that this has been done by Google. So you might think that you need the power of Google and the expertise of Google. But what I will show you in the following of this talk is that these tools, they are now at reach for almost everybody. We did start 3 years ago, and I will show you what we have been able to do in learning in a very short time and to apply it to our data. So this was the introduction to show you that we shift from observation with camera, observation made by camera that are analyzed by computer, and now let's say that the algorithm they face a limit somehow, and this limit has been overcome with the deep learning. So I will explain now what is deep learning. But actually, this morning, Christophe Zimmer made a very nice talk to explain in much details what is deep learning. In my talk, I will give you a lot of reference. This one is the course, the lesson by Yann Lequin at the Collège de France. He's one of the pioneers. He's the French pioneer for the deep learning. And in 2015, he made a talk at the Collège de France when he has been introduced. I remember the talk by Christophe. Deep learning showed up in 2012. And 2015, the guy he entered the Collège de France. It's no mistake here, I guess. So this is a very nice course that you can look on the internet. But myself, I didn't want to make my talk on the explanation of the deep learning. So it's very nice that this morning Christophe made it instead. What I want to tell is the way I feel when I do deep learning. Me, I think that I feel when I train neural network that I am a Pokemon trainer. This is what I'm thinking every day. I'm training neural network, and I'm thinking I'm just a Pokemon trainer. And actually, I made a slide of it because it means that anybody, myself, you, if you use data, you can train a neural network to perform a given task. So this is really the main thing about deep learning is that you are not doing any coding, you are not doing any hard coding of rules, of software to obtain a very good result. You just need the data. And then you train the neural network to achieve these results. So what has been explained in detail this morning is that you have pair of image, let's say, thousands of such a pair of image, and then you train a neural network to be able to transform this image into the target. And at some point during the training, actually it managed to have a very good fit to the data. And this is the function, this is the neural network, this is the transformation, this is the model that you use later on. So to get this model, which can be considered as an algorithm, you just need the data and the training do it for you. Sorry. And then in the field of microscopy, this morning you have heard that deep learning showed up in the 2012. In the field of microscopy, it showed up in the 2018. The first paper will really show how powerful it is to analyze or to make some post-processing in the field of microscopy. It appears in 2018. And this is one of the pioneer paper that has been shown already by Christophe this morning. It allows to denoise a picture. So on the left, you have a very noisy picture. It is taken at a low exposure time. You have the image that is obtained at a long exposure time, so with much less noise. And then you train a neural network to go from this image to this image. And the result is this. That means that now we have a neural network which is able to transform this image, very noisy, to this image. So you think it's fancy, it's nice, but actually it changed the whole field of microscopy because now we can make pictures at a small exposure time, low exposure time, so that the sample is not distorted by the flux of the light. It will not, and it just changed the way we can do microscopy now. We can make very fast images, and then we treat after all to get the nice resolution. And actually, Christophe is one of these authors of such a paper which appeared also in 2018. And these two papers show that it's possible to change the microscopy itself, to make a short time exposure, and to put a neural network to increase the resolution or the contrast, etc. So we can see the deep learning as a microscope on steroids, if you want. I'm really stuck with this. That's another example where they do virtual staining. That is, you take an image that is in gray level because it is not stained. So a tissue, a cell is basically transparent. If you do not put any dye, any staining, you will get such a picture. It is gray. And to see in details the nucleus and the tissue and etc., there are some dye or staining here is a colorant that is used on a daily basis to do tissue staining for an pathology. And here, they train a neural network to go from this gray level to this color image. And it just works fine. And what you have to realize here is that you don't need annotation, manual annotation here. For the example I've shown you so far, what you need for the data are only microscope images. Here I take an image in gray level, I take an image in color, and then I can train a neural network. It's not just like when you have to make a classifier for dog and cat. You need to make manual annotation 10,000 of times with some humans who have to point whether it's a cat or a dog. Here there is not such a problem. You just use the microscope images. So this is very In the field of microscopy, this is very usual. This is another example. So this is the input of the neural network. Again, it's gray level images. And they train the neural network to stain in blue the nucleus, the DNA. From this image, they can go to the membrane, the cell membrane image. From this image, they can go to the nuclear envelope. And usually, this is done with different dye that you have to put on the cells, and you have to use a fluorescence microscope that is quite costly. This microscope to make this gray level image is a regular bright field microscope. So basically, you could tell, you take a bright field microscope, this is cheap, and then you add some neural network to get the result of a fluorescence microscope that would cost 20,000, 40,000 of euro. I'll just show you what has shown up in the year 2018. This is another group, they just did the same. Gray level image of neuron, and then you have the true image that is obtained with a fluorescence microscope. And now you train a neural network to go from the bright field microscope to the fluorescence microscope. And this is the result. So there is a network, it is able to convert this image into this image. But again, so far, it's about observation. Now we observe the image, we are not measuring. Now I will go to the measurements. Now you have this image, and you want to make the segmentation. So what you do is that you show, during the training of the network, an image with the segmentation. But here it's not the same thing. You need a human to make the annotation of every single cell. So here it's quite painful. If you want to train a neural network to do a segmentation task, there's no other way than making by hand all this segmentation. And this paper will show up in 2021. They make the manual annotation of 1.6 million of cells. So they paid people to make the segmentation, but they did not trust much these people. So there was on top another panel who checked that the people have made the annotation correctly. But doing so, if you are able to have such a large data set, you are able to perform a very good neural network for segmentation. This is another segmentation algorithm that is from 2021. It has been trained with 70,000 of cells, manual annotation, and this one is considered today as the best algorithm for segmentation. So I've shown you the field of deep learning applied to microscopy. This has occurred within 4 years only. It changed the way we observe, and it changed the way we measure. But now there are some questions that you can raise. Is it available? Because I've shown you a result that has been obtained by Google with 200 engineers. But can you work with Google? But I will show you that now, from now on, it is all open access. But the main question is it reliable? If you, for instance, you go from this bright field image to a perfectly an image of a fluorescent dye that really stands the chromosome perfectly. Do you trust this algorithm? You have to raise the question, can you trust this? It works perfectly, but can you just trust it? Another question is, can it let discover? If you use the data that has been processed by a neural network, will it be able to make any discovery? Because basically a neural network, it reproduces what it has seen many times. But will it miss the things that it has never seen? And you can think that what you want to discover is something that has been never seen before. So if you go through the neural network, you can ask the question, will you still be able to discover? I will show you that there's other way to go for the discovery through a neural network. And the question, is it alchemy? This is a question, or is it a black box? What is this thing, actually, deep learning? So I will not answer this question. These two questions. For this, I will show you what we are doing now, and you will try to figure out if we do answer this question. With what I am doing in my group, do you think we are discovering anything, or do you think it is alchemy? Let's say you will answer this question by yourself. I will just go quite fast on these two questions. Is it available and is it reliable? So I will show you the literature that is showing up in 2021. So it's nice to see that deep learning, it changed the observation, 2018. Then it changed the analysis to 2021. And now people, they ask questions. And they find solution to use it and to make it to get the best of it. So is it available? No, it's fully available. In this paper, there is a list of different algorithms for segmentation. This is all available. You can put your picture on the net and get the segmentation of your picture. And there are many of them. There are many platforms, and it's working fine. And there's another paper here. It's not only about the segmentation. It's for all different tasks. So this is segmentation, this is denoising, this is image transformation. This is fully available through this platform. So if you want to get into this neural network and give a try, you can go for these two papers, and they will show you the way to use this neural network. If you are a biologist, this paper is very nice because it gives a large overview of the use of deep learning for the biologists. And it shows that it's not only images, you can apply a neural network for the protein. So I advise you this paper if you're a biologist. And the question is it reliable? And you see that this is addressed in 2019. And in this paper, they show that, of course, deep learning is very powerful to analyze the data. But there are also some pitfalls. And there are three of them that has been, and the terminology is the following. There is the hallucination problem. If it was a classical algorithm, you would say there is a bug. The hallucination problem is that the neural network is just not working. There are many reasons it is not just working. It is because the data set that is used for the training was too small, or it was too far from the neural network was too small itself. So basically, the larger the data set, the larger the neural network, the better are the results. But if you do not train properly the network, it might just fail. Generalization problem is another problem. It means that it was discussed this morning, your neural network has been well trained on a given data. But then if you apply it to another data that is close but slightly different, it might not work well. So this is the generalization problem, but it is not just for neural network. This is true for any kind of a classical algorithm. You can optimize your algorithm for given data, and then it might not work for another data. So this is not only true for deep learning. And the adversarial fragility problem, it was shown this morning, you can tweak a little bit the input, and the neural network will give a completely different output. So this is more a security issue here. And I will give you some example. So these are the results of different neural networks that are applied for this image here. It's a noisy image. And you see that this first neural network is working fine. It is able to make the denoising, and you check it with the high signal to noise ratio image. So this one is worked fine, because actually it has been trained on this kind of image, where you see actin within the cells. This is the result of a classical algorithm. So you have to realize that the deep learning is not always better than classical algorithm. So there is the question, when do you use a classical algorithm instead of a deep learning, and vice versa. But here is the generalization problem. That means that this neural network here, it has been trained on another kind of image. It has been trained on DNA images that has nothing, these are still cells, these are still biological samples, but it's too far from these images. And you see that now the algorithm is not working at all here. So this is the generalization issue. That's the same for segmentation. CellPose is the state-of-the-art algorithm, and it has been trained on different kind of data set. And if you use this model, that means that it has been trained on this model, you see that the segmentation is quite okay. It misses a bit the small details, but overall, no, sorry, this one is good. This one is good. He finds every different cells. He makes a good separation between these two dividing cells. So the neural network trained with this model is fine. But you see that another neural network with another model is not fine at all. The result is not convincing here. So the main discussion in this paper is that you cannot buy a neural network. You have to test it anyway. So you can find neural network on the net. You will have to test it. You can train your neural network. Obviously, you will test it. You can ask someone to train a neural network for you, but you have to test it. That's the main message of this paper. And you see, we are in 2021, so we could have told this much before, but never mind. And these are all the metrics that you have to compute to test a given neural network. So if your neural network is making cell detection, let's say, you have to compute the precision and the recall to check the efficiency, the score of the neural network. And this is painful here, because now you have to make a manual annotation to test your neural network. This is true also for segmentation. You train a neural network on your data, you have to make manual annotation of all the cells and to check it out. But this is compulsory. You have to do that. Otherwise, you're just using a neural network, and basically you cannot trust it. So this is the main message of the paper. And again, there are tools, there are open access tools to do this job. But for some neural network, you have to do manual annotation. You cannot just not skip this stage. So it is reliable, provided that you do the test. It is not reliable on its own. You have to test it. That's the main message from the one in the community. And this paper is also nice because he gives you some advice whether you should use a classical algorithm or deep learning for your application. So is it available? Yes. There are many open access. You don't need to code by yourself. You can train a neural network with it without any coding. You just have to send all your data to a cloud. So this gives a problem of security. Do you want to give all your data abroad? That's another question. But the training, you can do it on a server, or you can download the trainer on your PC, and you do the training on your own PC. So this is fully available, and you don't need to be an expert. The main message is really to test your neural network in the end. That's important. And it goes with the question, is it reliable? Can it discover, is it alchemy? I will not discuss this. I will just show you from now on what we are doing in my group. And in my group, basically, we have a goal. We want to obtain a global representation by neural networks of, well, let's say everything. We want to have a representation by a neural network of the macroscope, of the light transport, of the biological sample of its future. So we want to use the neural network to represent mostly anything, what we are working with. So it's an objective. We started 3 years ago, and I will show you what we are working on. We use a particular macroscope. It's a lens-free macroscope. So it's only an LED and a CMOS sensor. There is no lens, no objectives here. You put the cells on top of the CMOS sensor, and this is what you get. So what you get here is a diffraction pattern. You don't get a picture of the cells, and we have been working for years to develop an algorithm that is able to reconstruct the image of the cells from this diffraction pattern. So this is a lens-free microscopy. And the gray levels here, it's proportional to the optical thickness of the cell. So the brighter, the thicker the cell. And if you compute the integral over the cell, it's proportional to the dry mass, to the mass of the cell. And a mass of a cell is typically in the range of 200 picograms. So it's a microscope, but somehow it's also a weighing device. We are able to weigh the cells. And this is a time lapse. So if you put this microscope into an incubator and you put the cells onto the microscope, you are able to make a movie So it's a movie that lasts in real 3 days. And you see thousands of cells here, because there is no magnification on this microscope. So actually, the size of the acquisition is the size of the CMOS sensor. So it's 30 square millimeters, and you are able to look at up to 20,000 of cells. And this point is important, because if you want to train a neural network, you need a lot of data. And this microscope, actually, it fits quite well this deep learning thing, because you can make the acquisition of a very large data set quite easily. That's one thing. And of course, you can zoom in the picture. Here, what you see is a cell culture. These are adherent cells on top of a glass substrate, and you see there are some division, and you see there are some migration, and there's some interaction. And with classical algorithms, at that point there was no deep learning because we started 10 years ago. This is all done with classical algorithms. So now we are following up a cell in time. So there is segmentation. First we do the segmentation of the cell. Then we do a cell tracking to follow up the cell. And now we are able to plot as a function of the time the cell dry mass. So you see it's in picogram. For a given cell, we can measure the cell area, the thickness, the speed, and so on. So this is all done with classical algorithm. And now we have been using a neural network to make time prediction. So it's a paper by a PhD student at the CERA I'm working with. And we are doing time prediction. So this is a plot of a dry mass as a function of time. In blue, in plain blue, this is the input of the neural network. And we train a neural network to predict the mass of the cell for the next 8 hours. So we could use this to predict the value at the stock market and earn a lot of money, but we use it to plot the dry mass of the cell. And in the dashed line, this is the exact measurement. So you see that it's not that bad, it's not that good, but it is able to make prediction of the mass of the cell, meaning that the neural network is able to encode somehow the information to make a prediction. So at this point, the neural network is still a black box, but it is encoding some knowledge here. It is able to predict the dry mass over 8 hours. And we don't use that as a crystal ball to predict the future. Actually, we use this when it is not working. This is my point. This is the input of the neural network. And then we make this prediction in orange here. And then we make the comparison with the real data. And we make the difference. And we put a threshold. And when this difference, this integrative difference, is larger than a given threshold, then we look at these cells in particular. And this example, what you see is that the cell is dividing in 3. This is not possible. Well, this is what has been observed, but it's not possible. The cell always divides in 2. We have 3 cells here because first you see a fusion of 2 cells, and the one that has fused when it divides gives 2, and then you see the 3rd cell here. But what's important here is that doing so, by training a neural network that is able to predict or not the future, we are able to pinpoint something that is not normal. If you want to do this with hard coding, you have to define rules. You have to define all the rules of what is not normal with the respect of biology. And you have to put thresholds everywhere. You have to put parameters everywhere. And you have to optimize this parameter. Here, it's completely different. At no point we put knowledge in the software. We just train a neural network to predict the future. And when it is not predicting the future, it is actually pointing out a cell that is not normal. This is another example. Again, he managed to find another cell that is dividing in 3. And here, in this example, he finds a cell that is fusing. So 2 cells give 1 cell. This is not normal. So it is pinpointing not normal cell on its own. It's quite interesting. So it's another way to analyze the data without any priors, without any knowledge. Thanks to this way of using neural network, we are able to highlight cells, anomalies, only with the data without any knowledge. This is not a discovering thing, but it's a new way to analyze the data. Now we'll discuss quantitative representation. So I told you so far, we made all these steps with a classical algorithm. This is the raw data from the lens field microscope. We do some denoising, then we do the cell detection, cell segmentation. This is all done with classical algorithm. And if you are following well, you understood that from 2018, now we can replace all these tasks by a given neural network. So instead of this classical algorithm, you put a neural network, we put a cell detection network, and you put, and so on. You make a pipeline of different neural networks. And in the end, what the biologist or the scientist you want to get are the value, actually. You just want to know whether the cell has such a surface area, such a maximum thickness. What is important here are the values here. But then you have to train a network here, there, and there, and you don't get the results. You get a segmented image, and so you need another algorithm here to get the value. The point here is that we question whether we could just not replace all this pipeline with one single neural network. And to do so, we have to implement a new representation, and it's a heat map here. And for every single cell, we put a dot here, and the value of the dot is the surface area of the cell. Meaning that these cells have a surface area of 1,000, these cells have a surface area of 500. So from this classical algorithm, we compute this quantitative representation of the image. And now we train a neural network to go from this image directly to the results. So somehow it will compute all the pipeline, but with a single neural network. That's what we have done. And this is the results. Now we have this very large image with thousands of cells. And then we have a single neural network that computes directly the measurements. So we go straight to the results. It's a heat map of in this case, this was the cell dry mass here. But we have computed this for all the metrics, like the cell surface area, the maximum thickness, the sphericity, the length. And we have compared our results with the ground truth that is obtained with the classical algorithm. So again, we develop a neural network and we test it immediately against the data that has been obtained from the ground truth. And it works fine. We got a quite good linearity, meaning that with a one neural network, we can compute a lot of cell metrics directly. And the nice thing about this is that this neural network, it goes very fast, because what we did not discuss so far, one of the advantages of the neural network is that it can be computed very fast. In this case, in half a second, we can compute the result over 10,000 of cells. Whereas with a classical algorithm, for instance, only this step, the cell segmentation, can last about 5 minutes. So now we have replaced all the tasks by a single neural network, and the main advantage is that you can compute the result very fast. And for the microscopy, it's important because from now on, we will be able to take decisions. We can measure, observe, analyze very fast, and we can make decisions from now on based on this analysis that goes that fast. And an action could be picking up cells or modify cells that you have pinpoint with this algorithm. And we test it against noise, because you can tell it works fine if the image is clean. But we add noise, we add background, and noise, and the result still fits the ground truth. So now we have a new representation, meaning that we have this image, and now we are able to compute this image where every cell is represented by a matrix of values. In green, the surface, in yellow, the dry mass. And now we are shifting the representation. We are not looking at the... We might not look anymore at the image. It could be... it's questionable. But we can look directly at this image, which is a new representation, which is a quantification of every given cell. And here's a point that so far I discussed that we observe, we measure, we analyze, we make it faster and better with deep learning, but maybe we are going all wrong here. And this point here is that the interaction between the cells are more important than the measurements done on the cells. And the example given by this professor when he presented his work at the Royal Society is that if you study in very much detail a single termite, meaning that you measured everything about the termite, you know his past and maybe his future, you will never understand how the complexity of the termite emerged. So in our case, it means that you need to measure everything from the cells, but this is not the end of the story. You have to go to the interaction. And I will show you that interaction between cells is obvious. Over the years, with our lens-free microscope, we have measured many different phenomena. What you see here is a cluster of cells, and they exchange some cells from one cluster to another, and it goes back. So there are obvious interaction here. The same here. There are cells that are getting out of these clusters. They are making paths, and then the paths are making a network between the cells. So there are definitely interaction here. And you see, if you look at this on a greater picture, you see all the interaction here that are showing up in this cell culture. This is another example of a cell culture. What I didn't told is that this is recorded in 3D because this is a cell culture in 3D, and all this interaction you see them clearly once you go from 2D when the cells are only on the plane surface to 3D. There are much more interaction, and you see there are some mechanical force. So the interaction in some of them are mechanical, right? We have many of these examples. You see these clusters is not interacting, it is eating up the other cells, but this is an interaction here. And this one is a nice movie. You see the self-organization. These are endothelial cells, and all the cells are making a network themselves. So now if you want to model all this interaction, you can build a model, meaning that you try to define some rules, to define by yourself some laws. And I've done that. I made a model by myself. I'm a physicist, so I thought, okay, these are force of repulsion, force of attraction. And I fit this model to the data, and it works quite well. But it fits well to this data. And I've been working for weeks to adjust all the parameters and to let it fit to the data. But now, if I look at another phenomena, you cannot not apply my model anymore. You have to make a new model for this observation. And if you are a nuclear physicist, you will say, well, this is nuclear fission here. You have an explosion. The neutron is going to this other atom, and this is a nuclear fission. I will make a model with nuclear fission. So my point here is that we can, from now on, we can make model to study all this interaction, provided that you go to another representation, which is a representation of the cell culture as a graph. So you still need to make the measurement of every single cell, but then now to go further, you make connection between these cells. And now you are making a graph network. And this is the large view of the cell culture, with all this connection between the cells. And now we're going to study this interaction. Of course, we're going to study this through a deep learning method. And it's a new method that is showing up. It's a graph neural network. It has been developed to study the social network. So it is used to study the peoples, basically. A graph is a node. These are edges between the nodes. And usually, one node is one human, and this is another human, and there's a relation between the humans, and they want to know if you're looking to this movie, does the other guy look at the same movie? So this is for Netflix, this is for the Facebook. This is all developed on a nasty question. But we can reuse this to apply this to the cell culture, provided that you represent it through a graph network. So now, one node is a cell. Attached to the NEL, the embedding, the node, what is called node embedding, you put all the cell matrix. The edges connect the cells that are close one another, and you can also put embedding on the edges. This can be the distance here, how many times they touch each other, and so on. So you have a description of your cell culture as a graph. And now you are able to train a graph neural network that will make the transformation from one graph to another graph. In our case, this is the cell culture at time t, and this is cell culture at time t plus 1. And doing so, you are able to make a prediction. You are able to make the calculation that accounts for the neighboring, that accounts for how these values interact or how these values are correlate to one another. You compute all this information, it is aggregated, it is transformed, and then it is pulled back to these new values, and you can just train it, and you just get this neural network that is able to make the transformation here. It works very well on mechanics, on fluid mechanics. Where is the mouse? So here, every node is a droplet of water. The node embedding is the position of the droplet, the speed of the droplet, and the mass of the droplet. And you can train a neural network with fancy simulation that are very costly to compute. So this is the simulation, and you train a graph neural network to reproduce this. And when you reproduce this, you have a full model of the mechanics of the fluid. And this one is computed very fastly. The simulation, again, it lasts for hours. So you have encoded the laws of the mechanics of the fluid in these graph networks. But you already know the laws. So that's why we believe that it's important to address this methodology to cell culture instead, and try to, again, find laws here. And for this work, we are looking at a PhD student. So please apply. You can work for 3 years on this nice subject in Grenoble. We have just now, since yesterday, we're going to ski for the whole winter. If you like to ski and to do some deep learning and look at cells and make microscopy, you are very welcome to join in our group. So we believe that this new representation as a graph network will change the way we do analyze the data. It can allow us to make a new classification of the cells, unsupervised. That means we do not make any annotation. We can identify the cells that are very important for the structure. So these are regulatory cells, but conversely, it can identify cells that are very harmful for the structure, and this might be deviant and malignant cells. These are the features we would like to extract from this graph neural network representation. I don't know how many times. 10 minutes. You have more than 10 minutes. Sorry? You have a little more than 10 minutes. Okay. Question included? Or? Including questions. So I will give some time for the question. I will show you a last thing I didn't expect to speak about, but I want to go a little bit further to show how we use, in a different way, the neural networks. I want to show you that a neural network can represent the object itself, can represent the cell itself. And we use it to reconstruct it in 3D. So this is the last application, last example that I want to show you. And the point here is the representation. Here we have constructed a neural network that can represent the object. That means that we take, here it's a worm, it's a C. elegans worm. We cut it in slice, we cut the object in slice, and all the layers, the blue layers, are the refractive index of the object. And between the layers, there is the light transportation. So we have given a defined structure to the neural network. It's not a regular neural network. It's a network that is made of index of refraction, light transport, index of refraction, light transport. And it represents the object. It is the object. And when we train this network with some acquisition, as the input we put different light, different angle of illumination. At the output, we put the acquisition. And now we train the neural network to reproduce these results. And actually, he managed to reproduce these results. You show them a lot of images, and he's able to go from another light direction. I can tell you what is recorded on the camera. So that's one thing. But when he had managed to make this transformation, actually, he has recovered the object itself. because now it means that it has put weight on all the voxel of the refractive index. It has made the light transportation from one layer from the other. And the point is that when you train the neural network, you recover the object. Usually a neural network, it is used to make a function, to make a transformation. Here we use this representation of a neural network to tell that it is actually the object. And when we train it, we open the black box. Usually you don't open the black box because you are happy with the results. Here we open the black box, and when you open the black box, you get the object in 3D, in full 3D. So in blue, you have the value of the refractive index, and in yellow, you have the light transportation from one layer to another. This was during the confinement, so we get some data from the UC Berkeley of the worm elegans. And this is the reconstruction. When you open the black box, the neural network is the object is a neural network, and you have a complete adequation between the two. When the confinement was over, we built our own microscope in a few minutes. And then you see here the different LEDs to get different angles of illumination. And we have been able to reconstruct, this was a mouse embryo in 3D. So this is a neural network. This is a mouse embryo, but this is a neural network. So it's a fancy way to use neural network. And this is why I told you that we would like to have a new representation brought by deep learning. So this new representation, these are cell time prediction. I told you about quantitative representation to get the measurement straight from the image. And I told you that from now on, we are working with a new type of graph network to look for the interaction and not only the measurement within cells. And with this slide, I end my presentation, and I will take some questions, please.
Very interesting stuff. Thank you. What are the cell types that are in all those images? You're sort of quantifying all these cells, and what are they?
Yeah, there are many of them. This is a mouse embryo. The ones that are moving from one cluster to another, these are prostatic cells. This one, they are endothelial cells. So these are the cells that are making capillaries. So these are endothelial cells, and this one are prostatic cells, this one. And when we do adherent cell culture, usually we look fibroblast cells, HeLa cells, also endothelial cells, stem cells. We look at pretty much a lot of different types of cells, right?
About the graph network topic, I understood that the idea was to predict what the next state of the graph.
Yeah.
So don't you think that it could work as well just to take a movie and from an image at time t to predict what would be the image at time t+1? And what do you think would be the difference by taking the two approaches?
Yeah, it's a good question. Looking through this graph representation, what we want to figure out is how the values are connected to one another. For instance, we want to figure out, but without any priors, if there is a connection for the dry mass. Let's say what we want to figure out is if the dry mass is very high here, does the dry mass of the neighboring cells will be low, or will they get, or maybe they will try to get larger than this dry mass? This kind of relation between the values themselves, this is what we want to figure out. And to do so, we have to integrate the interaction between the cells. So that's the main idea of the graph network. Conversely, if you just look at the images, somehow there will be no point of to try to predict the next images because we just have to record it. We record it, the next image, and we are done. And we have tried to predict from one image to an image, and actually the neural network is not that good for that because somehow he will average all the possibilities. Meaning that if the cell is going up, if the cell is going down, or if the cell is going left or right, he will make an average of this. And somehow you will get a blurry image because you make an average. So when you do time prediction on the image itself, it's not that powerful. And again, if we want we just need to make the next image, and that's better. There have been work right this week where they make interpolation between images, and it's interesting because you can increase the frame rate. So interpolation between two images is interesting because you can record at a frame of an image every second, and you can get a movie with hundreds of images per second. So interpolation is maybe. So this is my answer. This idea is new and is not very sounded. It's not very well. We did not thought much about it, but. I took the opportunity today to present it and to give a thought about it one more time and to get some question here. But this is really new, and this is not. This has to be done, right?
Maybe I can ask a question about this also. So you compared this with the example of the water cube that.
Yeah.
So I guess in your particle-based simulations, the laws of physics are actually encoded in the simulation.
In the simulation, right?
In some ways, but not in the neural network.
Exactly.
It's able to do it fast because it learned, but it doesn't know anything about the rules.
It doesn't know anything about the rules.
Somehow I thought you wanted to use this for this cell network to actually understand the rules of interaction between cells.
Yeah.
Can you actually do that? Because the fluid mechanics example doesn't do that.
Yeah. So if you take the fluid mechanics example, it works provided at some point you have given the position of the droplet, you have given the mass of the droplet, you have given the direction of the droplet, and then it works. You have to give some values to the droplet here. So if you remove one by one the different metrics of the droplet, you can figure out what are the important metrics.
You can do correlations.
You can do correlation.
To figure out what is important, but you don't get the rules.
You don't get the rules, but that at that point you get already something.
Sure.
So that means that if you're able to make prediction with the dry mass and the cell surface area and the motility with these three values, it means that with these three values you can explain the data. That's the first thing. But next, to get the rules, that's the goal, and that will be the Graal. We still have to develop tools, right?
But even for simpler like fluid mechanics example,
can
you get an Navier-Stokes equation from neural network?
Yes, they have done that for physics. They have done that for the solar system.
Okay.
Meaning that they figure out that you just end up with two latent variables that were the momentum and the radius. And from the neural network, they have been able to extract the physical laws for the solar system.
Classical mechanics, which are
simple.
Okay.
And they make very simple system like a pendulum and etc. So it shows that you can.
Could they have predicted completely different rules also because they know the result?
Exactly. So this is what they are doing now at the CERN. They want to reanalyze all the data with this perspective to find something else than the standard model, because the standard model is working perfectly fine, and they have time now at the CERN because they closed down for years. So. But that's the goal at the CERN. There are people working on that, try to reinterpret the data without any priors and try to find new phenomena and maybe new laws. So this is my answer to can it let discover? We don't know at this stage. So far, we have rediscovered the mechanic laws, but maybe I think it's worth to give a try with a biological system. That's my point here.
Any other questions? I don't think so. Thank you very much.
You're welcome.